Hello everyone, it's my honor to share with you one of the recent progress using machine learning algorithm to identify lineage assignment change of SARS-CoV-2 on the PangoLEARN system and identify important mutation and their interactions. So let's review the nomenclature system for SARS-CoV-2. Before the outbreak, we have a GSET system, which stands for the Global Initiative on Sharing All Influenza Data. This system is characterized by using some major mutation and compare them with the reference genome to identify some large clades using simple and quick methods. Followed by the next strain system. By the name, you can know they actually follow the conventional phylogenetic and phylodynamic analysis to identify evolutionary stable clades and subclades for not only influenza but also pathogens. So after the outbreak, we now have the PANGO system, which stands for Phylodynamic Assignment for Named Global Outbreak. It also takes into account the epidemiological relevance, not only rely on evolutionary relationship, which I will illustrate later. So let's take a look at the illustration of this nomenclature system. The WHO label system is the world-known simple system using the geek alphabets that people know which virus we are facing nowadays. As you can see, the next-gen clade system also have different naming system, very different from the Pango lineage system. If you take a look at their trees, you identify that these systems are not usually agree with each other. As you can see nowadays, we are having the Omicron XBB system, which can categorize into 22F system in the next-gen system. Now, let's take a deeper look at the PangoLEARN system, which is based on the PANGOLIN system. The PANGOLIN system is designed to classify genetic lineage for SARS-CoV-2, and they have to use the dynamic nomenclature system. They initially trained the model with a conventional maximum likelihood-based assignment algorithm, which is used for conventional phylogenetic tree development. And then in July 2020, because large amount of genomes are identified and submitted to the global system, they have to shift to the classification tree to speed up the classification process. And the last version of the Pango system, which is PANGOLIN 4.0 system, they adopted the random forest to classify the lineage in a more efficient fashion. So that's why they call it dynamic nomenclature system, because they have to update their model on a monthly basis. You can probably guess that their model is not able to predict or forecast the lineage, but just to classify the lineage according to our knowledge. So what's the criteria for new or updated lineages? Of course, we have to start from the genomic evidence. So all the lineage is expected to share at least one single common ancestor, and they often represent as a monophyletic or paraphyletic clades. There has to be distinguishable by at least one unambiguous events, which is such as the single nucleotide change, insertion deletion, or recombination events. And they also have to have at least 5% or 5 sequence high genome coverage to know that they share the same parent lineage. Another important feature of the PangoLEARN system is they also have the ability to use the epidemiological significance to assign new or update the lineage. So the epidemiological evidence includes the events in new country or regions, or new waves in the same location, or a lineage have uniquely increased or higher growth rates compared to the other lineages. And they could also have a very different phenotype. Say they have the higher transmissibility, they are causing more people admitted to hospital or ICUs, or some immune escape feature. They could also represent to cross-species transmission events, or they carry a specific biological interest of concern. Therefore, this is their limitation. It sometimes couldn't reflect the real epidemiological or genomic evidence of the new lineage. In the recent publication in Science, they identified that some lineage classified by the PangoLEARN system have multiple successive peaks in some regions. So let's take a closer look at this paper. The MIT MGH group, they developed a pi-R0 model using hierarchical Bayesian multinomial logistic regression model. And they trained with a very large database with 6.4 million SARS-CoV-2 genomes. They used the surrogate outcome, which is the growth rates, as the virus fitness, and they give the outputs of the posterior distribution of relative fitness. So they successfully refined 1,500 Pango lineage into 1,300 finer clusters. And then in this figure, they successfully identified BA.2 as having almost 9 times higher fitness than the 8 lineages. But they also have some limitation. First, this is the most arguable distribution of the Bayesian methods, followed by the higher order interaction that is not able to be checked in this relatively simple model. Lastly, although they have a very large database, it still has the source of bias. The data-intensive region could be contributed to some rich countries that have more sequences than the other parts of the world. And the Chinese, the researcher also developed their own system called the Mutation and Variance Monitoring and Pre-warning System. The objective is very Chinese, I would say. They hope to establish mutation blacklist and whitelist. In this study, they also used a large database containing 6 million SARS-CoV-2 genomes. Interestingly, they used the term de novo mutation, which stands for a clade with more than 50% mutation in this study. And among the 6 million genomes, they identified 296,000 de novo mutations. Then they performed the association analysis between, again, the growth rates and the de novo mutation. They also have to use a relatively simple method, which is the linear least square regression in their study. So they identified the blacklists include mutation in some receptor binding regions of the spike protein, in some protein coating region, which could mean premature termination codons, or for some important truncation that would result in the fatal phenotypes, or even for some non-synonymous mutation in the coating region that could result in more hydrophilic amino acid residual, or may have affects the structure of the corresponding protein and the consequent gene function. And interestingly, some mutation was never found in other regions or other genomes, which may mean that this mutation is fatal to the virus, so that resulted in distinction of the virus. In terms of the whitelist, they identified 185 de novo mutation in every part of the virus. Conventionally, we always pay attention on the coating region, but these whitelists are also noted in some non-coating region, as well as the cross-chromosomal distribution, or RBD region, spike M and M protein. They are identified everywhere in the virus. Just to let you know, for some resource-limited country or era, the government or the corresponding agent is able to sequence only the spike protein. In other words, we will miss other important mutations. So these methods also have some limitations. First, again, is the growth rate does not always representing the fitness in all lineages. And then is their simple linear least square regression methods. First is the similar linearity issue, and then is the capacity to identify the interaction between the genes. So why is that so important to understand the mutation? First is to understand the biological meaning for transmission or pathogenesis. And then, of course, we always hope to predict important phenotype of new lineages. Lastly is to identify functional and epidemiological importance of this mutation. For example, the D614 gene is associated with higher viral load and was once treated as the candidate vaccine antigen. And for this mutation, they're believed to link to increased transmissibility, enhanced binding to ACE2 sites, and they might be associated with antibody escape. And then the gap in knowledge to understand this important mutation is to identify functional important mutation in the context of large background of genetic variants with very little or even no phenotypic consequence is still very challenging to researchers. So our objective is to prove the concept of using machine learning algorithm to identify important mutations, and then doing the cluster analysis to understand the possible interaction between the mutations. And we want to have a higher resolution by looking into single lineage instead of whole picture. So let's take a look at the outbreak in Taiwan this year. As you may know, before this year, we've had very few cases in Taiwan. And this year is particularly interested because the lineage BA 2.3.7 is very special cases in the world. As you can see in the world map, this lineage happened mostly in Taiwan, but also could be found in the rest of the world. Let's take it deeper into a look into the CoVLineage.org website, and you can know that we have some assignment in this lineage. And this is again not important events in Taiwan, but also important lineage in the rest of the world. In another study published by the National Taiwan University Hospital in Hsinchu branch, as well as the collaboration between the Chang'an University, they sequenced 1,900 sequences, and they also downloaded about 900 sequences from GSAT, the public database. And they zoom in to take a closer look at the BA 2.3.7 to understand the events in the cluster. So they identified the index case from Malaysia, which is collected in March 2022. As you can see in the different color represent the different geolocation. There are different groups of the location can be identified. And then also identified a very unique signature mutation in this lineage. So we take the advantage of this lineage and we downloaded both BA 2.3.7 and the parental lineage, which is the BA 2.3. We then use the PANGOLIN system to understand how these lineages are updated in their system. In other words, why the PangoLEARN system failed to identify them before the outbreak. We then use the machine learning algorithm to identify important mutations. Lastly, we use the cluster system to understand the interaction between the mutations. So let's take into account a deeper look into the different lineage distribution. First is the BA 2.3. Most of them stay the same and not updated in our databases. Then we can see the most BA 2.3.7 comes from BA 2.3, and some of the BA 2.3 comes from BA 2. And the distribution of this lineage is very interesting in the world as well. As you can see here, the most lineage update happened in Asia, but it also happened in the rest of the world. After collected data, we have to pre-process data in order to be able to fit into our machine learning algorithm. So first, we need to digitize the sequence. We adopt the DNA walk methods, assigning different base pair into different numbers. Then we follow the PANGOLIN or PangoLEARN procedure to do the QC check as well as the alignment. Now this is an illustration of how the PangoLEARN system is able to do the QC and the following procedure. Once we have the input data, we first do the QC to check the empty sequences, to check the N contents, and then we write the final file that contains the correct sequence. So after the QC procedure, we have to align to the reference. And then we check if the sequence are correctly aligned, then I store the aligned or assigned sequence into the output. Then we report it back to the PangoLEARN system and then finish all the procedure. So back to our procedure for the machine learning algorithm. In this study, we adopted a sequence to image method in order to take advantage of the deep learning method. This method is also could avoid the alignment issue often caused by the reference sequence. As you might know, many phylogenetic analysis of SARS-CoV-2 still use the very original Wuhan strain as the reference sequence. That might cause the bias since the pandemic outbreak has been going on for 2 or more years. It could also be used for the de novo exploration, and the methods are pretty much mature and available, such as the Deep Insight, Deep COVID-19. And lastly, we could use the transformed data to take advantage of the deep learning methods. Then let's take a deeper look at the procedure. Very similar to our previous algorithm, we first download the database from both databases, and then we try 2 methods. One is the k-mer preprocessing, the other is codon indexing. And after encoding, we do the image transformation using the TSNE methods. And this method could generate a filter and we convert the sequence into the image. As you can see here, the 4 coarse illustration from alpha, delta, beta to omicron. As you might be able to identify the difference of this image, the machine can also identify the difference of this image. So what's the TSNE methods? This is a relatively popular method nowadays, using the t-distribution and do the neighboring embedding methods. As you can see in this animation, you just shrink all the distributed data back into their cluster, and then we can try to identify what are these clusters. And TSNE methods also have different optimization methods. For example, the Barnes-Hart method is one of the methods to identify the clusters. So how well are these methods on our database? Again, for variant prediction, including alpha, BM, beta, delta, eta, gamma, iota, lambda, mu, and other, you name it, a cluster. The MLP methods after the non-k-mer methods has a relatively good accuracy both on training and testing database. So let's go back to our original plan, is to compare different machine learning algorithms to identify lineage update, which is our outcome in terms of the new lineage in the database. As you can see here, although it's blocked by my video, the XGBoost has the best testing AUC. Here is 0.9519. Comparing to different methods, including the CNN method followed by the image transformation methods. So again, we proved the concept that deep learning are not necessarily by default the best methods. And then after the random forest model, we identified top 20 important features by looking into their mean shape value. On your left-hand side is the average shape value, which is actually a gaming theory generated value. Try to identify how much a model will change after taking a single feature out of the model. So as you can see here, because we only have binary class, they're having the evenly distributed color. The V21A51A is the most important mutation identified in this model. On the right hand side, you can see some of the mutation has positive effect, but some has negative effect. How could you interpret the effect? First is the x-axis, stand for the shape value again, is the impact on model outputs from a single feature. And the scale here just represent the value of the feature because it's of the our encoding methods. We only have two kinds of possible feature value. But you still can see that some are having very complicated distribution. For example, the 2156T, the red color distribute on both sides of the shape value. So we still need to do a correlation analysis in order to understand their interaction in this random forest based model. So then we identified top 5 important features from the previous feature ranking algorithm. As you can see here, again, the spike protein gene K97E, this is already transformed to the amino acids mutation sites, and the N440K is the important gene on the spike gene region. On the open reading frame region 1A and 1B, we also identified 3 important mutations in the model. As you can see in this figure, again, 2 mutations in the spike gene and 2 in the open reading frame 1B and 1 in the open reading frame 1A region. We further try to see if they have any corresponding evidence to support our finding. We found out that in one publication that K97E is associated with severe pediatric cases, as some of the audience might still remember that in the beginning of the outbreak in Taiwan, we found that some kids suffer from this pandemic outbreak. And the N440K is a very interesting gene. This is very highly variant gene, and there's a lot of reports not only in the BA 2.3.7 but also for other genes. And interestingly, the L631F mutation is very unique mutation in BA 2.317, although it's not published in any peer-reviewed manuscript, but only reported by our CECC in Taiwan. Lastly is these two mutations, and again, very interestingly, the importance is not yet identified by other body of evidence. So in conclusion, we proved the concept that machine learning algorithm can help to identify important mutation before many evidence accumulated. And again, deep learning is not necessarily doing better than the other machine learning algorithm, and future deployment is still possible. As I have limited time, I didn't have time to illustrate how we check the interaction between the genes. And last, I want to give my acknowledgement to my research team, and these are the lab members in our team, as well as to our colleague at Hopkins and our sponsor in NIH. Thank you, and I'm happy to answer any question that you might have.